Welcome to MLT 240, Special Clinical Microbiology. In this first section, we're going to cover some basic parasitology, and we're going to cover the first section that covers the amoebas that affect humans. In this section, we are also going to be covering collection of specimens for parasitology and examination of those specimens. So what is a parasite? A parasite is an organism that lives on or with another organism or a host at the expense of the host organism. So what is the impact of parasites? We're lucky here in North America in that North America does not suffer from many parasitic diseases, though many other parts of the world have high levels of parasite-induced morbidity and mortality among humans, animals, and crops. And when you hear morbidity, you should think about the state of being unhealthy, and mortality as the number of deaths or relating to death. The World Health Organization names 5 parasitic diseases among the 6 most harmful diseases afflicting humanity today. About 4.5 billion people harbor parasites. The total population of the world in 2021 was 8 billion. It's estimated that 489 million people have malaria, and it kills 1.5 million people each year. 25% of the world's population, that's about 1.9 billion people, suffer the consequences of parasitic infections, and these consequences include malnutrition, iron deficiency, anemia, and other chronic health effects. Vaccines are in various stages of development for several different parasitic diseases. Our classification system with parasites is the same as with other organisms. We start out with kingdom, phylum, then go down to class, order, family, genus, and species. You can see some of the different kingdoms and phylums that we will be covering of the different parasites throughout this semester. I'm not going to spend a lot of time going over all these right now, but we will be covering a lot of these throughout this semester. As we talk about each of the different parasites this semester, we'll also be talking about the parasite's life cycle, and a lot of the parasites will be accompanied by this chart or graphical display that helps you to understand the transmission and the spread of infection and how that occurs. The life cycle also displays how the location of the parasite stage in the human body correlates with clinical symptoms and pathology. There's typically 5 parts to the life cycle of our parasites. The first one is the location of the parasite stage in the human host. The second one is the means by which the parasite stage leaves the human host. Then there's when the parasite is infective, or the parasite stage that is infective to humans. And fourth, the means by which the new human host is infected. And then the fifth one is the site of development and maturation of the parasite in humans. There's some basic terminology for parasitology that you need to be aware of. So let's quickly run through some terms. There's an accidental host, which is where there's an infection of a host other than the normal host species. There's the definitive host, which is the animal in which a parasite passes its adult existence or sexual reproductive phase in, or both. There's the carrier, which is a host harboring a parasite but exhibiting no clinical signs or symptoms. Commensalism is the association of 2 different species of organisms in which one partner is benefited and the other is neither benefited nor injured. Ectoparasite is a parasite that is established on or in the body surface of a host. Endoparasite is a parasite that is established within the body of a host. The host is a species of an animal or plant that harbors a parasite and provides some metabolic resources for a parasitic species. Infestation is the establishment of an arthropod on or within a host. Symbiosis is the association of two species of organisms exhibiting metabolic dependence on their relationship. Obligate parasite is a parasite that cannot live apart from its host. Facultative parasite is an organism capable of living an independent or a parasitic existence. So this is not an obligate parasite, but potentially parasitic. And then a vector is an arthropod or other living carriers that transport a pathogenic microorganism from an infected to a non-infected host. So a lot of times for our parasitic samples, our sample is a stool sample. So a couple things that you have to keep in mind with our stool samples: they have to be collected prior to radiological studies. If barium sulfate is given to a patient must wait 5 to 10 days before submitting another sample because it can affect the sample. Also, if a patient is been given antibiotics or taking antibiotics, you must wait 2 weeks after treatment is ended to provide a new stool sample. It is preferred for a parasitic sample that the stool sample is naturally passed. The container for the sample should be cleaned, and it should be a wide mouth container. We do not want any contamination of urine in the sample. The container should be properly labeled, including the patient's full name, the hospital or other ID number, date, time of collection, time the specimen arrived to the lab, and the label must be on the container, not just the lid. The reason we don't want just the lid labeled is because if that lid gets separated from the container, then you will have an unlabeled container. So preferably label the container and the lid, but at very least label the container. For our specimen collection, we usually do multiple collections, usually 3 every other day over a 10-day period. As far as timing to examine our specimens, if it's a liquid specimen, if the stool sample is more on the liquid side, it needs to be examined within 30 minutes after collection. If not, trophs will revert back to the cyst stage, and you'll understand what we're talking about as far as trophs and cysts. That's the stage of development here in a few minutes. In semi-solid stool samples, you want to examine within 1 hour of collection. If it's form stools, you need to examine within 24 hours of collection. For your specimen collection, we a lot of times use preservatives, and we use a 2-vial system as the standard means for transporting specimens intended for ova and parasite examination. 1 vial is going to contain a fixative, and it's usually SAF or PVA, and the other vial is going to contain formalin. Okay. Also, samples can be preserved by refrigeration or MIF. IMF both fixes and stains organisms, and it's usually suitable for concentrated or concentration procedures. The formalin is an all-purpose fixative, and then the PVA and the SAF are both suitable for making permanent smears. You can microscopically examine stool samples as well. For each specimen, you can do a direct wet mount on a fecal sample, or you can do a concentration technique or a permanently stained technique and examine under the microscope. This is an example of someone doing a direct wet mount. The consistency is important for determining the type of organisms that can be found in the sample. A mushy or liquid stool, you might see trophs of your intestinal protozoa more commonly. Because they do not survive long, you need to examine in a timely manner, like we were saying a while ago, in the previous slide, you need to examine within 30 minutes. Form stools, you may see protozoan cysts more frequently. In liquid or form stools, you may see helminth eggs or larvae may be found in either. Adult worms in stool, if you want to see those, you may need to break up a sample with a applicator stick to observe, to be able to see the adult helminth or worms. In samples, you may see blood and mucus. You might see bright red that indicates acute lower GI bleeding, or bloody mucus, which suggests ulcerations. And this material should be examined for trophs. This is showing you a little bit more in detail on how to make your direct wet mount for a soft or a liquid stool sample. And this is a rapid screening technique to determine if you have trophs in a sample. You can also use iodine to help you visualize eggs or cysts, but it will kill trophs. There's two different fecal concentration methods that we can use to recover parasites. One is the sedimentation procedure, and the other one is the flotation method. In the sedimentation method, if there's any eggs present in the sample, they should be down here in the sediment of the sample. In the flotation method, this is where you use a liquid that has higher specific gravity than the eggs or cyst, and the eggs are going to float up to the top if there's any in the fecal samples. So all the debris is going to go down to the bottom, and all the eggs are going to float up to the top. This method is not used, or should not be used, if you're trying to recover aperculated eggs. Aperculated is eggs that have like a little lid, because if the little lid comes open on the eggs, then those eggs will not float. Also, just something to note, if only a single concentration procedure is used, it should be the sedimentation method and not the flotation method. The last procedure for microscopic examination would be, you could do a permanent stain. There are various staining methods that can be used on parasitic examples to see different protozoa. And some of the stains include the trichrome stain, the modified trichrome stain, and the iron hematoxylin stain. Other diagnostic procedures for parasitology include the cellophane tape test for pinworms, and that's over here in this image. That's simply where some tape is stuck to the bottom of the child that's suspected to have pinworms, and when the pinworms come out, then they stick to the cellophane tape, and that tape can be stuck to a slide, and those pinworms can be observed under the microscope. There's a acid-fast stain, a modified Kenyan acid-fast stain, that's used to confirm things like Cryptosporidium. We can use blood smear preparations and staining for blood parasites, for the recovery of things like Plasmodium. And usually you want to prepare a thick and thin smear when you're doing those. And they can be stained with a gypsum stain. There's the knot technique for concentrating microfilaria. And there's also some culture media and agar plates that can be used for parasitology. And then lastly, they have immunodiagnostics and molecular testing that includes PCR testing and ELISA testing for parasitology. In this first unit, we are going to be covering protozoa, but this first section that we're covering in this lecture, we're going to be talking about the amoebas that affect humans. So what are some general characteristics of our protozoa? They're eukaryotic, unicellular animals. Most of them reproduce by binary fission. So what does binary fission mean? It means asexual reproduction. It means separation of the body into two bodies. In protozoa, life functions are carried out by specialized known organelles. Each species of parasitic protozoa usually is confined to one or just a few species of hosts. There's at least 30 species of protozoan parasites that affect humans. There's usually two major modes of transmission for protozoa. There's usually the ingestion or bite by an arthropod, and there's a few that's transmitted by sexual contact. And we will talk about the amoebas, the intestinal and tissue flagellates, the ciliates, and the sporozoas. But first off, in this particular section and lecture, we're going to be talking about the amoebas. So this is just to show you the basic structure of an amoeba. The amoebas have a nucleus, they have a cell membrane, they have a cytoplasm, they have vacuoles, they have these arms that they can move around by and extend out to engulf their food, called pseudopods. So this is just the general overall structure of what the amoeba looks like. Let's go over some general characteristics of the amoebas. Again, we've already mentioned they have pseudopods to help with their movement. They have two different stages, the trophozoite and the cyst stage. The trophozoite or the troph stage is the motile reproducing feeding stage, and it's mostly found in the lower GI tract in this stage in humans. The cyst stage is the non-feeding, non-motile stage that many amoeba form, that is the infective stage to humans. The usual transmission is by ingestion of fecally contaminated food or water. The number of nuclei and other structures differ from species to species of amoeba, and the number of nuclei aid in the diagnosis of which species a patient has. Here is the life cycle of the amoeba. If you'll look here, it starts with when the human ingests the mature cyst, and it goes through the body, goes down through the intestines, and then the mature cyst is passed through and out into a fecal sample, and it's going to reinfest another human when that fecal sample contaminates the food and water that another person comes in contact with. So let's go over some terminology that's just related to the amoebas. So axoneme is a bundle of microtubules associated with proteins that make up the core of cilium or flagellum. Okay. Axostyle is an elongated supporting rod or tube that runs the length of certain flagellate protozoans. Frequently, these project out the posterior end. They can be single or multiple. They can be filamous or rigid, and they may vary with the species, but they provide an endoskeleton framework, and they may function for locomotion as well. Chromatin, this is basophilic nuclear DNA. Cilia is multiple hair-like processes attached to the surface of the cell. Functions, usually for motility. Cyst, this is the non-motile stage that is protected by a resistant cyst wall formed by the parasite. In this stage, the protozoa are readily transmitted to a new host. The cytostome is the body of the cell apart from the nucleus. The endostome is a small mass of chromatin within the nucleus. It can also be referred to as the karyosome. The excitation is the transformation from a cyst to the trophozoite after the cyst form has been swallowed by the host. The flagella is the plural of flagellum, and it's the extension of the ectoplasm that provides locomotion. It resembles a tail, and it provides movement with a whip-like motion. The mastogophria in the flagellates is the subphylum of protozoa having one or more flagella. The pseudopods is a phototrophoplastic extension of the trophozoites of amoeba that allows them to move and engulf food. Sarcodina is the subphylum of protozoa, including all the amoebae, both free-living and parasitic. Trophozoite is the amoeboid vegetative asexual form in certain sporozoa. Monogamy is the development of only a portion of an ovum. Now we're finally going to talk about some of the different species of amoebas. We're going to talk about intestinal amoebas first. We're going to talk about Entamoeba histolytica. This is the major pathogen of the group. It causes amoebic dysentery and amoebic hepatitis. So there's liver involvement, and the most common and gravest complication is the liver involvement. It causes chronic infections that may last for years. It's transmitted by contaminated food and water. Humans can serve as carriers for Entamoeba histolytica, and it also can be transmitted sexually. And this particular amoeba has worldwide distribution. As far as laboratory diagnosis of E. histolytica, you want to collect at least 3 fresh stool specimens to be examined. Those stool samples can be permanently stained, or concentration methods can be performed on those stool samples. You can do a saline mount on those stool samples as well, but they must be examined immediately by a experienced personnel. You can also do antigen-based immunoassay testing and serologic methods for E. histolytica. When you're looking under the microscope, you have to be careful when looking at your cysts and trophs, because some cysts of other species are identical to the E. histolytica cyst, but you can differentiate if you have presence of trophs. Your trophs are usually going to have ingested red blood cells. So let's look at E. histolytica as far as the differences between our trophs and our cysts. You're may see some motility in our saline wet mounts with our trophs. You also are going to see those ingested red blood cells, and that's what you're seeing here. Those dark red spots inside the troph is the ingested red blood cells. You're only going to see one nucleus. This is the nucleus in each one of the trophs. The size is going to be anywhere from 10 to 20 microns, and the size is going to be elongated. Okay, or the shape's going to be elongated, I should say. In the cyst characteristics, this is the infective stage. You can have anywhere from 1, 2, to even 4 nuclei. In this example, you have 4 nuclei. 1, 2, 3, 4. You have small central karyosomes and uniform peripheral chromatin. That's what you're talking about. The central karyosome is right in the center of the nucleus, and you're going to have cigar-shaped chromatin bars. So this is what that's referring to right here in the image. And the shape is going to be round, and the size is going to be anywhere from 10 to 20 microns. These are our non-pathogenic amoeba, and we're going to talk about each one of these on our next slides. This is E. coli. This is Entamoeba coli. This is the amoeba. Don't get this confused with E. coli, the bacteria. So let's compare the trophs versus the cyst. The troph only has one nucleus with large irregular shaped eccentric karyosome. So the karyosome is off center. See how this is off? It's not completely in the center of the nucleus. It usually has sluggish motility. It exhibits a granular cytoplasm. It ingests bacteria, and you're not going to see any red blood cells like we saw in E. histolytica. The size is less than 10 microns. For the cyst stage, it's large, eccentric karyosome, and you're going to see coarse, irregular peripheral chromatin, and you can see up to 8 nuclei. This example has 1, 2, 3, 4, 5 nuclei. You may see chromatin bars with splintered ends. I really can't see any chromatin bars in this particular example, but the shape is going to be round, and the size is for the cyst is also going to be less than 10 microns. The next species is E. hartmanni. So let's compare the troph to the cyst. You have only one nucleus with a small central karyosome. The karyosome is here, and that's a bundle of chromatin right in our nucleus. You have no cytoplasmic inclusions. The size is less than 10 microns, and these trophs are usually irregular in shape. For the cysts, for E. hartmanni, they're usually small. You have central karyosomes with even chromatin. You can have anywhere between 1, 2, or 3 nuclei. In this example, I see 3. You can have cigar-shaped chromatin body with rounded ends. So there is representing the chromatin body. The size is less than 10 microns, and the shape of the cyst is usually round. Usually in a microscope, you want to have an ocular micrometer to get the measurement right to help identify E. hartmanni, because this is a smaller amoeba. So always remember this looks very similar to histolytica, but this is always smaller. So when you hear E. hartmanni, think small, less than 10 microns. E. nana is up next. The troph has a large karyosome, and you're only going to see one nucleus with no peripheral chromatin. It has the large irregular blot-like karyosome. So this big dot right here that kind of makes the nucleus look like an eye, this is your large karyosome. The size for the troph is 6 to 12 microns. The cyst form can have up to 4 nuclei. They're large, irregular, slumped karyosomes with no peripheral chromatin, and usually they're oval in shape overall, and the size can be anywhere between 6 and 12 microns. Next is I. busheli, or Amoeba busheli. The troph has large irregular karyosomes with no peripheral chromatin, and it can be confused with E. nana. It only has one nucleus. This is the karyosome, and the size can be anywhere from 8 to 10 microns. The cyst stage has a large glycogen vacuole that stains brown with iodine. This is that large vacuole. So this is the telltale sign that you're usually looking at I. busheli. You only have one nucleus with large irregular karyosomes and no peripheral chromatin. Okay. The size is usually 8 to 10 microns. Next is Blattocystis hominis. The troph versus the cyst form. The troph is rarely seen, so we're going to focus more on the cyst form. The cyst is anaerobic. It has questionable pathogenicity. It has a round central large vacuole that takes up about 90% of the organism. You can see anywhere from 2 to 6 nuclei that are usually dark staining surrounding the vacuole. You use saline in processing because water can destroy these cysts. The size is anywhere from between 6 and 40 microns, and these may be confused with yeast cells when seen in a sample. And you usually use the trichome stain to ID these cells. So if you're looking over here at this particular image, this is a trichrome stain. And if you're trying to decide, is this a cyst or a troph, you need to look at this red structure within the organism. This is the vacuole. So if this is the vacuole and it looks like it's taking up most of the organism, what do you think it is? Which stage? You should be thinking this is the cyst, because remember that large vacuole takes up about 90% of the organism. Now we're going to move on from our intestinal amoebas to our opportunistic tissue amoebas. These can be found in fresh and salt water, or maybe even moist soil or decaying vegetation. There's two of these. There's Naegleria fowleria and Acanthamoeba. Let's first talk about Acanthamoeba. It can cause a chronic form of meningoencephalitis or granulomatous amoebic encephalitis, or GAM. It can cause keratitis associated with poor contact lens care. Infections occur through inhalation of contaminated dust or aerosols, or through the invasion of broken skin or mucous membranes. Onset of symptoms usually are slow, but can be even weeks or years before symptoms appear. As far as our lab diagnosis and findings for Acanthamoeba, there can be an examination of CSF or cerebrospinal fluid for trophs. Brain tissue can be examined for trophs and cysts. There can be corneal scrapings done for eye infections. The tissues can be stained with cauliflower white. There can be histologic examinations of infected corneal tissue. We can also do indirect immunofluorescence antibody testing, which is a ID technique of choice. And if you look here in this image, this is actually an image of brain tissue, and this is an actual troph of Acanthamoeba in brain tissue. This is just showing you what a Acanthamoeba cyst looks like versus the troph. The last species that we're going to cover in this lecture is Naegleria fowleria, and it occurs most often in summer months, and people usually come in contact with it in ponds and lakes where Naegleria is free living. It gains entry through the nasal passage and migrates along the olfactory nerve. Within several days, it invades the brain. It can tolerate high temperatures and chlorinated water. There's usually a rapid and fatal course within 6 days, so diagnosis is often made at autopsy. So this is a pretty fast-acting parasite. It causes PAM, or primary amoebic encephalitis, which causes rapid brain tissue destruction, headache, fever, nausea, irrational behavior, coma, and then death. Here is the life cycle of N. fowleria. Again, the person is usually in contact with water. The water goes up the nose, it crosses over the olfactory nerves, and it enters the brain tissue. As far as the cyst and troph form, we really don't know anything about the cyst form, but here is an image of the troph form. And you guys may have be familiar or have heard about this amoeba before, because this is usually referred to as the brain-eating amoeba. And this one we heard about a few years ago with the Whitewater Center down in Charlotte when some of the Olympic athletes were training down in Charlotte, and this amoeba was found in the water down there. As far as the distribution and prevention for Infallaria, the distribution is again found in warm water sources like lakes, streams, ponds, and swimming pools. It can even be found in mud puddles. The prevention and control is adequate chlorination and medical, community, and public education. Also posting off-limit signs to known contaminated areas. This concludes our first lecture for MLT 240. If you have any questions or need further clarification on anything, don't hesitate to reach out through email or during lab time, or come see me during my office hours.